to make diagnosis. So I need PCR to help me amplify that particular portion of DNA that I'm interested in. Amplification, you know what that means, like making it, like you say something, you want to amplify something, like you want to make several copies of a particular I have few material in a sample. I want to make several copies of it. So it's a technique that we use to make these copies by using our cycler, and then we are able to make sense or diagnostic sense of what we have. So method that is used when there is a requirement for more DNA that may be in a typical sample. So you may have a sample and you have few DNA, but the diagnosis you want to make requires you to have a lot of DNA, right? A lot of copies of that DNA. So you need to make several copies of it by cycling it, maybe 45 times, 36 times, depending on the number of cycles you are going to be running with your PCR. So next, please. And also it allows the amplification process to be limited to specific target. So what this does for us is this. If I have a stretch of DNA, right? You know, in DNA, there's what we call introns, there's exons, there's poly A stretch, and there is a lot of a portion of DNA that we call junk DNA, the portion of DNA that does not code for anything. So, but for the introns and exons, the coding and the non-coding part. So for the coding part, we have genes in that region. Genes are the ones that code for the activities that we perform, right? Your ability to see light, your ability to taste, every of the activity you perform, right? It's the genes that are responsible for all of these activities. So when genes are translated to RNA, you know, these translated genes are then converted to form proteins. Abi, in our, if you understand the central dogma, we are getting there in the course of this lecture. So the method that we use when there is requirement for DNA more than is necessary, that's the first one. But it allows the amplification process to be limited to specific targets or specifically targeted segments of the DNA. So the coding part, for instance, I have an organism in a sample, like let's say malaria parasite in a sample. And I understand that there is a region in malaria parasites that maybe allows this organism to enter or gain access into the human cells, right? So, and I am able to identify that region. So I can make primers if I want to make diagnosis. For me to know that the parasite is present, I will do targeted amplification of regions that are specific to that parasite that will be in that sample. So if those regions are not gotten from the amplification, then I can say, oh, this organism is not present in this sample. Next slide, please. So sample processing for PCR, it is very important that we take sample processing serious for PCR. Like every other laboratory procedure, you know that the reception of sample in the lab, you know, tells a lot about the quality of results you are going to get. So a sample coming into the laboratory with wrong sample bottle, you know, it has impact on the results you get. And then the data or the information of the patient that you are going to be getting. So also is PCR sample coming into the lab. So there are some acceptance criteria for us to say that a particular sample is fit for testing when we are accepting samples for PCR. So one of those is the sample packaging. We are talking of a very powerful technique. Remember I said you have a sample and few copies of DNA, and this technique can amplify these few copies of DNA to millions or billions of copies. So let's say I have several hundreds or several thousands of DNA in a sample. The technique is so powerful that after 35 cycles or 45 cycles, you can be having up to billions of copies of that specific DNA. So it's a very powerful technique. So if I have these samples plugged together in a cool box, for instance, I have 10 samples, and one of these samples is positive. right? And then I pack all of these samples, 10 samples together in the same Ziploc bag. And one of them that is highly positive or positive spills. And then it has access to, even touching the cover of a negative sample. Because of this powerful ability of PCR, or the method that we use in PCR to make several copies. So that few copies of DNA that may contaminate a negative sample can make it become positive, and you are going to be having ambiguous results. So sample packaging is very important when we are considering sample acceptance that we use for molecular testing, especially in PCR. So how do we package samples rightly? We normally advise that sample be packaged singly. So I have sample in EDTA container, right? So I'm going to have to put this sample, if I'm doing a batch transportation of sample, like I have more than one sample. So all I would simply need to do is, let me put this EDTA container in a universal bottle, wrap a cotton wound on the neck of the sample bottle, and then I cover it tightly, put it in a Ziploc bag. Then I'll treat every other sample in that batch the same way, and then package everything together, and then I'll send everything to the lab. So I'm not going to mix up 10 samples in a Ziploc bag and send to the lab, simply because if anyone that is positive leaks off and then contaminates a negative sample, you are going to be having ambiguous results. And the next thing we can also talk about is anticoagulants. The preferred sample that we use, especially for diagnosis, when we are talking of blood, diagnosis that has to do with blood. Remember, it's not only blood that we use for PCR, right? You can use swab, you can use dry swab or wet swab or anything. You can even use soil sample, you can use any type of sample for PCR. But when you are talking about blood for diagnosis in a clinical setting and you want to do PCR, the preferred anticoagulant is EDTA, right? Reason being that most of the other anticoagulants that we have, like heparin and heparin and fluoride oxalate, some of them are known PCR inhibitors. So when you use sample bottles containing some of these anticoagulants, they compete or bind selectively to DNA polymerase, and then you will see that you are going to be having false negative result at the end of the day. So the best anticoagulant that we use is EDTA. And then we have transport temperature as something very important too. If we in our history, when we started the lecture, I gave a breakdown of the structure of DNA that was described by Karimulis, right? And I said they described DNA as double-stranded, spiral staircase structure. So you see that double strand of DNA gives it a very stable form. The two strands are held together by hydrogen bonds. So there is this bonding like that makes it very stable. So DNA is stable. But remember, it is not only DNA that we run PCR on, right? We are going to get to that. Even though PCR is designed to run, is designed to use a DNA as the template, but we still do PCR using RNA as a template. So in this respect, we have to be very particular about how we transport our samples to the laboratory. That's especially for those that are having samples coming in from different centers, Now, why am I saying this in respect to transport temperature? If your samples come in hot, for instance, RNA is not a stable genetic material like you have for DNA. So RNA can easily be denatured. So when you have samples coming in without cool box, samples coming in not on cold chain, right? And maybe the sample had to travel a long distance to your laboratory, you can be having false negative results. If you have some positive samples, they could come out as false negative, especially when RNA is the material of interest in that sample because it is not as stable as DNA. So we normally advise that if you are having samples coming to your laboratory and you want to use it for PCR, it should come in cold chain, especially if the distance that the sample is coming from is quite far from your laboratory. So the next thing is the biodata. Like I've also earlier said, for instance, most of the investigations we do or most of the testing we do with PCR are things that could cause a lot of legal issues. For instance, a sample is coming into your lab for paternity dispute resolution, and then in the molecular lab, and there's a part where you have to do some form of amplification. That's why I'm capturing that as an example. Imagine treating such a sample and you are mislabeling one person's name for another person. The legal implication, what that can cause. Apart from the fact that it could break homes, you know what it can cause when the people find out that you actually exchanged names. So it is very important that all of this information be looked at. So especially as we said that samples are coming in double packaged. You have the primary bottle and then you put it inside the universal bottle. Sometimes you are going to be seeing one name on the first bottle and another name on the second bottle, possibly because some people want to reuse bottles. So the universal bottle they used to transport one sample before, they now put the other new sample inside it. They recycle it and put another fresh sample inside it, and it's coming to the lab, and you are not careful enough. You just write the name on the outside, not knowing that the name inside the original sample bottle is a different name. So biodata and getting patient information is very key in accepting sample into the laboratory. Next slide, please. Okay, so sample processing for PCR, DNA and RNA isolation. The sample has come to the lab. We accept the sample into the lab if the sample meets the acceptance criteria that we have just discussed, right? So how do we go from here? How do we process the sample before we can use it for PCR? The acceptance criteria, like we said, it must come in the right temperature. It must have come in the right sample bottle, and then the biodata must be completely filled and all of that. So now you have accepted. What next do we do? So the next step is DNA and RNA isolation. So we isolate DNA or RNA, depend on what we want to do, depend on the downstream application that we want to perform. So we understand that, okay, for instance, the COVID-19 virus, coronavirus really brought to light the PCR, COVID-19 virus or the SARS-CoV-2 virus is an RNA virus, for instance. HIV virus is an RNA virus. Hepatitis B virus, we know, is a DNA virus. YC is an RNA virus. So depend on what you want to do, that will determine the type of a nucleic acid isolation technique you are going to use, whether to isolate DNA or to isolate RNA. So we have different procedures for DNA extraction, and we have another different procedure for RNA extraction. So the next step when we have accepted the sample is to isolate the nucleic acid material of interest, whether DNA or RNA. And like I've said, what informs that choice is the type of organism or material you want to work with, right? So first thing we do is lysis and inactivation. So in most cases, you know why we have to lyse the samples. Now, DNA, if you, in our cell biology, is inside the nucleus of the cell, right? And the nucleus is an intact environment inside an intact cell that is in the internal environment of an organism. I don't know if we get that analysis. Nucleus is an intact environment. That means it has its own membrane bounding it, separating it from the interstitial fluid, right? And then the cell itself is an intact environment. It has its own membrane, right? So for me to get access to the DNA that is inside the nucleus, that is inside the cell, packed inside the cell, right? I have to be able to break open these barriers or this membrane, right? So what we do first is lysis, in the sense that we are going to lyse the cells, right? And then we say lysis and inactivation because we need to inactivate the cell and lyse the cells and break it open. So when we lyse, we are actually releasing the nucleic acid material inside the cell to the pool of reagent. So what we use in lysis are set of detergents. It contain detergent, contain EDTA. We call them lysis buffers, lysis buffer. So when we apply this buffer, we apply samples to this buffer, there is rapid release of the nucleic acid material into the pool of reagent. So we call that lysis we do. So after lysing the cells, like I had the reagent that I have applied the sample and the cell is lysed inside the sample, right? Now we are going to have a lot of impurities inside that reagent. These impurities like your collagen, hematine, if it's from soil or environmental sources, a lot of impurities will be in that reagent. So I'm going to be looking for a way to
I can't see the screen. Oh.
Yeah, hello. Hello.
Good
Hello,
evening, sir.
everyone. Okay, yeah, sorry, a call just entered and removed my network. Hello, Mr. Israel. Hello.
We can hear you now, sir.
Yeah, I need the Dr. Israel to share the screen. Hello. I'm not sure let me chat
Yes,
him up.
I'll do that.
Okay. Sorry, it's a call that entered my phone and removed me from the group.
Over.
Okay. Yeah, thank you, sir. Sorry. Okay, so like I was saying, I was talking about lysis and inactivation. And I said the cell itself is an intact environment. The nucleus inside the cell is also an intact environment inside the cell. So for us to have access to the DNA to use for our testing, we have to first release all of those. And I said we apply lysis buffer, which are made up of detergents and EDTA and some other components, buffers and all. And then we lyse the cells, we inactivate. And I said there will be a lot of impurities, right? It is not only the nucleic acid materials that will be inside that solution that we have just lysed. So you have other cell components, right? And most of these cell components, some of them are known PCR inhibitors. And then even the EDTA and the detergents you use can inhibit the activities of DNA polymerase, which is the main actor in PCR amplification. So once they inhibit the action of DNA polymerase, you are not going to have amplification. And if there's no amplification, there will be no PCR. So we want to now find a way to make sure that it is only the clear or the only, we are only going to isolate a pure form of DNA or RNA from the sample. So that's what we now do next. And that's why we do, we now continue to precipitation. Precipitation is just simply addition of ethanol to that solution. Ethanol will help us to release the nucleic acid material. It's a kind of floating it. We precipitate and then we continue to other extraction and purification. So there are different procedures for extraction. We have extraction, automatic extraction procedure, whereby you have machines that could help the extraction to happen. Just add your sample and this thing that you have constituted, and the machine gives you the pure form of the DNA or RNA, depending on what you are isolating. So that's automatic extraction, that you use automatic extraction machines. And there are other methods of extraction that we have. We can have the a magnetic bead extraction method, whereby we use some magnetic beads in the reagents, and we use magnetic bead separators to isolate our DNA or RNA. We can also have spin column based extraction procedure. There's a module where you are going to be taught on extraction. I think that's if you have not done that already. So there's also spin column based extraction procedure. So all of this process is just for us to get the pure form of DNA or RNA that will not be having other impurities that can inhibit the activity of DNA polymerase, which is a key actor or key player in PCR. And after extraction, we do elution. Elution is, releasing only the pure form after all of the extraction procedure. And then we now do quantification, which is, setting up which is, checking the concentration of the DNA or RNA that we extracted. Next slide, please. Okay, so this is just a picture I placed here to show us how all of this process work. So this one is spin column based, right? Like I said, you can have the magnetic bead separators, spin column based, or you can have you do automatic extraction, depend on what you want to do, and depend on the downstream application that and how pure you want it to be. Now, look at this first tube here. Let's say that that purple thing inside the tube is our sample, right? We put it inside the reagent that you can see, the yellow one, right? So now that reagent is like our lysis solution that is going to lyse the cells, right? So when it lyses, we transfer it to a spin column. The spin column has a collection tube under it. So this spin column, you can see the blue part of the spin column. It usually contains silica gel. Is a silica gel membrane. So the silica gel membrane, when we transfer all of this liquid to the spin column, silica gel membrane will help us to form a bond with your nucleic acid material, whether DNA or RNA. And only the liquid that you use in lysing will come down through the gel, and then the DNA or RNA will stick to the membrane of that gel. So you now add other reagents to purify it, like kind of wash it. So in all of this, you centrifuge in between. So centrifugation will allow the reagent to pass through the filter of the membrane while the nucleic acid material sticks to it. So you follow all of this procedure until the last point where you can get your pure nucleic acid material. Next slide, please. So the next thing we do when you have gotten your pure form of DNA or RNA is what we call master mix. Master mix simply means putting all of the components or the reagent, PCR reagents together, right? So the component of the master mix, like you can see here, water, like bivalent cation, like magnesium ion, Taq polymerase or Taq DNA polymerase, DNTPs, template DNA, and primers. So master mix means all the materials, the components that are essential for a PCR reaction to take place, that will be in the PCR tube that you are putting inside the cycler. That's simply what master mix is all about. Next slide, please. Okay, so this is just explaining all of the components, one after the other. Component of PCR master mix, template DNA. The template DNA is that DNA that you extracted, right? The one you have purified, extracted, and purified that will be inside. So it's like your sample that you are setting up, right? So a template DNA, if your template DNA is present, and then the template, is specific, or the primers you design, has the template, that's when amplification will happen. So template DNA polymerase reads and copies the template DNA. So when we are talking about PCR amplification, in the first place, what is being copied, or we said PCR is like molecular photocopying, where you can make several copies of few copies of DNA that are present in a sample. So the copies that we are talking about that were originally present is the template DNA. So it is from here DNA polymerase can make several copies, and then this is what we design primers to target. For instance, the COVID virus, we want to target if the virus is present. We know that the virus has spike protein. So we design primers that can target spike protein. We design primers that can target the viral RDRP specific to SARS-CoV-2. We design primers that can target the N gene specific to this virus. So all of these are present in the template DNA. So it's the template DNA we make primers to target, and it is the template DNA that is also selectively amplified. So the quality of the template DNA is very important, like you can see here. So it must be intact and it must be pure. Remember, I kept saying you have to get it in a pure form. You have to get it in a pure form and it must be intact. It must be intact. Let's look at when we have a sample being transported in a very hot condition. DNA or RNA can be denatured, so it will not be intact. If it is not intact, you are not going to get good result, or you get no result at all. If it is not pure, those impurities that are going to be, if you don't have a good extraction procedure and it is not pure, then you are going to be having things that will competing with DNA polymerase during the reaction. And you are also going to have ambiguous results in the process. So the quality is very important. It must be intact, and then it must be very pure. And then the concentration, it must have a good amount of concentration. And then next slide, please. Okay, so the other component we are looking at is the primer. We have actually talked about primer, in a way. Primers are short fragments of synthesized DNA that bind complementarily complementarily to template DNA, right? So we know the law of base pairing from previous lecture, I suspect. Now, we know how the nucleotides pair with one another. So now I have an organism, and I know that, okay, let's say the gene I am interested in copying with PCR is A, G, T, C, G, G, C, G, and all of that, the way the nucleotides read. So primer, the way the primers will be designed is something that can complementarily bind to those nucleotides, right? So that's the primers that will be designed. Primers will be very specific. They are designed to be specific, and they are designed to anneal to that target alone that they were made for. Primers cannot cross-anneal. They cannot cross-anneal with any other primer they were not designed for. So in essence, it means that if I design primer for organism A, it cannot target another organism and amplify with another organism if I have followed all the rules of primer design correctly. And usually primers are short short sequences, like I said. They are usually between 18 to 22 base pair in length. If you have primers that are too long, there's something we call hairpin formation. There's something we also call primer dimer. You have to follow the law of primer designing very, very strictly. And then we have the fact that they are in pairs. We have forward primers and we have reverse primers. Please, I do not want us to actually bother about all of these details. We are actually giving you these details because we are professionals and we need to have understanding of what happens in the reaction tube. What you are seeing here is the PCR reaction tube, right? And the things that are inside. Because normally the manufacturer makes a reagent, right? You want to make a PCR. You are not going to say, oh, let me search for primers, let me design primers, let me synthesize primers, let me get the bivalent cation, magnesium ion, and measure the quantity I will use. No, all of these have actually been incorporated in the reagent you are purchasing, right? So all you need to do is to make sure that your reagent is maintained in the proper temperature. You bring it to room temperature, and then they can tell you, okay, add 15 microliter of reagent A to 5 microliter of reagent B. That's actually all you are going to see when you want to run this PCR. But just for extra knowledge, I have to bring out all of these components in those reagents so that you understand what is actually going on when that PCR is running. Next slide, please. Hello. Oh, thank you. Okay, so the next one is next components of that PCR setup are the nucleotides. We call them DNTPs, usually obtained from, the nucleotides. They are monomers of DNA. Remember, like I said, those nucleotides, adenine, A, T, G, C, that you used to see written in stretches when we are writing the figures of DNA, right? Single nucleotides are part of this setup. So we call them DNTPs, deoxynucleotide triphosphate. So they are part of the setup. So they are actually the building block. You know, I've talked about PCR, DNA polymerase being the one that synthesizes new strands of DNA. So these are the like blocks. When you are making a building, you know, it is the block, the bricklayer will be putting. So in this case, I want you to imagine DNA polymerase as the bricklayer, right? DNA polymerase is the bricklayer, but the blocks that is necessary to make the structure to stand are actually the single nucleotides. So they must be part of that reagent. Next slide. And then, okay, so the next one is DNA polymerase itself. DNA polymerase, like we have said, I said is like the bricklayer that is building the structure itself. While the DNTPs are the building blocks, the bricks that will be set in the first place. So I just want us to have a look, a little more understanding on this DNA polymerase. Usually, the machine that we call PCR machine, you know, we call is actually called a thermocycler. So the thermocycler works on the principle of, you see it increase in temperature, hold, decrease, increase, hold. It's just increase and decrease in temperature, increasing, holding, and decreasing and increasing, depend on what the program is set to be. And usually, you are going to be having temperature as high as 95 degrees Celsius. And then it will bring it down to 55 degrees Celsius, hold for some minutes, raise it to another maybe 62 degrees Celsius, hold for some minutes, and all of that. You are going to be having changes in temperature within the cycler, the thermocycler, right? And for our information, PCR is an enzymatic activity. Is an enzymatic reaction, is enzyme reaction, right? Now, we know what the properties of enzymes are. We know the optimum temperature with which enzymes operate, Abi, and we know that enzymes need cofactors to operate, right? Okay, that's sorted. Now, DNA polymerase as an enzyme from the name polymerase, we know that we have to selectively get a kind of DNA polymerase that can stand that kind of increase in temperature without being denatured. Enzyme activities, we know they are affected by temperature, Abi. So in this case, what we use in PCR is called Taq DNA polymerase. It is the only DNA polymerase that can withstand such high temperature. So usually it's gotten from a bacterium called Thermus aquaticus that is isolated from warm spring. So that's why it is suitable for this kind of reaction. We use Taq DNA polymerase as the major source of DNA polymerase for running PCR. Taq DNA polymerase, you can see some of the properties on the screen. It has speed. It is heat stable. It is a heat stable enzyme. It has speed and fidelity. The processivity, you know, it has the ability to complete long reads and the ability to read GC-rich templates. Next slide, please. Okay, so other components like the buffer, you know, the buffer supply optimum pH for the reaction, bivalent or divalent cation, as you may have to call, like magnesium ion, serve as cofactor for the enzyme activity. I told you the major enzyme here is DNA polymerase. Though you may have other enzymes that will also play a role, like RNA polymerase or whatever, depend on the type of PCR you are running. And then water is also a component in the PCR setup. Next slide. Okay. Next, so that we can just, let's try to round up this session. Next slide, please. Okay. Gene amplification by PCR is modeled after DNA replication. Next slide. I want us to see this slide very well to have a deeper understanding of what happens in PCR. You know, PCR is amplification of DNA, making several copies of DNA from a template DNA, Abi. In our own system, right, in the human body, we have DNA, and we have DNA DNA replication happening always in our system. Even as we are talking, DNA is replicating every minute, every second of the day. DNA is always replicating. Cells that are dying, you know, new cells are being formed always. And then DNA in those cells have to be the same with the original DNA from the old cell that is dying, right? So there's always replication of DNA. And this is the process that is listed here. And these are the enzymes or the things that are involved in the replication of the DNA that happens usually. So for initiation, we have DNA helicase, single-stranded binding proteins, topoisomerases, and in elongation, we have primase, DNA polymerase, DNA clamp, and then we have termination stage, DNA ligase, and then telomerase. Next slide. Next slide, please. Okay, next. Okay. Next. I think there are 3 stages. All right, thank you, sir. Okay, so I said DNA or the PCR cycling steps is modeled after DNA replication, like I just showed you, right? Initiation, elongation, and termination. So this is just like an example of what we just explained with what happened in the human body. But here we have the first stage as denaturation, right? Denaturation is when that double strand has to be separated. Remember, the double strand, like I said, in the double helical spiral case, according to Watson and Crick's description, has the coding region facing each other, right? And they are held together by bond. And the information you want to copy or the information you want to amplify is not just exposed. So it has to break, the bond has to be broken and the 2 strands separated for that portion you are interested in to be exposed. So this is where denaturation comes in. So in denaturation, we use heat to separate the double strand of DNA. And usually the heat that we apply is usually around 94 to 95 degrees Celsius. So you have one double strand of DNA. After denaturation, it becomes 2 single strands of DNA as you have in the screen. So in initiation, right, I'm just comparing it to what happens in the human body when DNA replication is happening. Helicase, as an enzyme, like you saw in that previous slide, helicase as an enzyme help us in separating the DNA strand in the portion that wants to be copied, right? But here we use it in denaturation, and it's usually around 94 or 95 degrees Celsius that we use. So the double strand of DNA becomes two single strand of DNA. And the next cycling step is annealing. Annealing is that stage where the temperature lowers to around 40 to 60 degrees Celsius, sometimes 62 to 72 degrees Celsius, depends on the reagent or the enzymes you are using, right? Now, in annealing, what happens is simply primers binding to the template DNA. And remember, the temperature I'm saying is between 40 to 60 degrees Celsius. Some will tell you between 62 to 72 degrees Celsius. You know, it depends on the reagent you are using, and it depends on the target that you are actually looking out for. So here, primers bind to their complementary sequence on the target DNA. Remember what we said about primers, that they are very specific. They are designed for specific targets. And if the target is not in the sample that you have extracted, okay, let's say you have extracted a sample and you actually did DNA extraction. Human DNA is in that sample, right? It will be abundant because it is human sample. And you are looking to maybe isolate maybe the herpes simplex virus. And you are thinking the DNA is also in that sample. So the primers you are designing is to target that particular virus, right? Now, the viral DNA is present in the sample and it has been denatured. That means it's now two single strands. So at this stage, when the temperature lowers, the primers will look for the specific target in that virus they were designed for, and they will go and attach to that target if the virus is actually in that sample that you added. But if the virus is not there, the primers will only float freely in the reagent. There will be nothing for them to attach. I want us to get this part very, very well. If the virus or the target, what you want to diagnose, is not present, the primers will only float. There will be nowhere for them to anneal or attach to. And then the next stage is extension and elongation. Or you say you could just, is extension and elongation. So in extension stage, after primers have annealed, DNA polymerase will now use the building block, which are the single nucleotides, or the one we refer to as the GNTPs, right? It will now start to attach them from the edge of the primers, and new strands of DNA will now be formed. So you now have, after extension, you now have double strands of DNA again, like you had from the initial stage before denaturation happened. And the DNA you are going to be having will be exactly the same thing with the original template DNA. Next slide. Next slide, please. Okay, so the chain cycle of PCR, denaturation. So all of this we have said, denaturation, extension, denaturation, annealing, extension, and elongation is called one PCR cycle, right? It's one PCR cycle. And usually in PCR, we could have 45 cycles, we could have 40 cycles, we could have 35 cycles, depending on the kit that you are using and how the kit was designed. So after all of this is just one cycle. So after one cycle, if I have one DNA template in my sample, after one PCR cycle, it will become two. And then after two PCR cycles, I'm going to have exponential increase. There will be doubling effects after every PCR cycle. So that's how we get to have our hundreds of thousands of billions of copies of DNA that we are going to be having at the end of the reaction. Next slide. Okay, so this is also an explanation. This is explanation of the heat profile. So you can see here, sorry, okay. So you can see here, this is an example of a PCR program. I had a template DNA at the bottom, double-stranded. You can see increase in temperature to around 95 degrees Celsius. That's denaturation. So it separates into two strands. Then the temperature comes down to around 50 degrees Celsius. You can see the primers annealing to it. And then there's a little increase in temperature to around 72, where extension and elongation will happen. And then you now have two double strands of DNA. So this is also one PCR cycle. Next slide. Okay, so this is just showing how PCR works, the exponential increase in the original template. You have one copy starting it. You can see the original strand, original template on blue, while the new strand is indicated by red. So it started with one copy. Next slide, next. And you can see how it continues to increase. Next. So after every cycle, you are going to be having double of what you had from the initial cycle. Okay, next slide. So it will still keep going like that, and every other cycle you have doubles what you have from the previous cycle. Next slide. Okay, just a graphical representation of how the increase happens. Next. Next slide, please. Okay, so with what we have done so far, I think we have been able to establish what really happens in PCR, what we do in PCR. We have been able to establish the fact that PCR does targeted amplification of DNA. What I mean by doing targeted amplification is that we are not interested in the whole stretch of DNA. We are only interested in targeted areas in the DNA that will help us to make diagnosis, right? So for instance, there's mutation in so and so gene in a person. You target that region in that part of the human DNA, and then you are able to establish that there is mutation. So you do targeted amplification and make several copies of that particular target. So that's what PCR does. And then we have been able to see the steps in PCR. We have been able to see what we do, denaturation of the template, annealing where the primers we attach, extension and elongation, and we're able to also establish how exponential increase of the template occurs after every cycle of PCR. All right, so now what we want to see next is the different types of PCR that we have. So we are going to be looking at different types of PCR in our next presentation. Okay, next slide. Next slide, please. Okay, next, just you can on everything down. Okay, so this first one we are considering is multiplex PCR. Multiplex PCR, according to, as the name may also already suggest to you, when you say something is multiple, right? Now, multiplex PCR help us to make several targets. We can actually do PCR and target several portions in a single DNA, or we target several portions in different organisms in a single sample. I don't know if we get this. We can have a single DNA That's DNA from a single organism. But we are interested in several targets, not just a single target. Okay, let's say I have a TB, a Mycobacterium tuberculosis in a sample, and I want to check if this TB, a particular tuberculosis is resistant to maybe different antibiotics, right? And I know that there are specific genes in this organism that code for resistance. And these genes code separately for resistance. So I'm going to be checking if a gene that codes for resistance to rifampicin or ciprofloxacin is present in this particular organism. So I can do a PCR to target for this gene at the same time in the same setup. Another example of multiplex PCR is, for instance, I have a sample from a suspected STI case, and there might be more than one organism or more than one thing responsible for that sexually transmitted infection. So I may be able to set up PCR and identify different organisms that this person is actually carrying. So multiplex PCR is a type of PCR that we do to have different targets in the same run. So we can set up PCR and say, okay, let's just diagnose if there's malaria parasite present, or let's check if there's Lassa virus present in this sample. But in multiplex PCR, we're actually looking at various targets. So even for the Lassa virus, we are going to be checking for different genes in the virus. You know, you are not just looking at one gene, but in the same setup. So it's not like you are going to set up PCR. Let's check if this gene is present. Let's do another PCR to check if this other gene is present before we can confirm that it is this organism. No, everything is actually happening in the same reaction, but you are having different targets or different organisms being identified in the same reaction. Next slide, please. Okay, so this other type of PCR, mutation-specific PCR, is a very important type of PCR in research. So for instance, I'm suspecting that, oh, this H2 HRRP gene. I think I should use this as an example. Most of people use RDT, right, to check for the presence of malaria parasites. And you see MP negative, MP positive. And the funny part is that the organism you are targeting and what the RDT you are using is actually trained or made to that target in making that diagnosis might have changed in this same organism that you think you are using it to target. So when we are looking at mutation-specific PCR, we are trying to find out, oh, at so and so region, is there a way we can copy this region of the gene and check if so and so mutation has happened in this particular region of the gene? So we are just targeting a particular gene, looking out for specific mutation. Has this organism changed its ability or its susceptibility to a particular drug? Has there been any mutation in so and so part of this organism that could predispose a person to so and so disease in the near future? Has there been any mutation in any site of this organism that could make this organism to be more virulent or spread faster than it used to be? So mutation-specific PCR is actually more of what we use in research to check for specific mutation in PCR. So like you can see on your screen, it detects the presence of a mutation or SNP, we call them SNP, in the region of interest of a gene, and it is selective. Selective amplification is achieved by designing primers that will match or mismatch one of the alleles at the 3' end of the primer, and it requires reaction to be carried out in duplicates. Next slide. So this next one is reverse transcriptase or reverse transcription polymerase chain reaction. Sorry, our time is far gone, so I have to just quickly run through most of these slides. So when we are talking about reverse transcription, we know what transcription is, we know what translation is, Abi. So transcription, reverse transcriptase or reverse transcription PCR amplify, isolate, or identify a known sequence from a cellular or tissue RNA. The PCR is preceded by a reaction using reverse transcriptase to synthesize cDNA from RNA, and it can be a single step or two steps reaction. So now we have PCR. Remember I said DNA is used in PCR when we started this in the introductory part of the lecture. PCR is designed to work with DNA. But now in the course of the lecture, we've been saying RNA, viruses, and all the likes of it. Now, we want to detect a disease caused by an RNA virus or RNA organism, right? Or want to check something that has to do with RNA. Does it mean that we cannot use PCR? This type of PCR, we actually got this knowledge from the RNA viruses or the retroviruses, sorry. We got this knowledge from the retroviruses, whereby they are able to, okay, let me use HIV as an example. The HIV virus, we know is an RNA virus, but is able to incorporate into the human DNA. So reverse transcription polymerase chain reaction is what we do when we want to work with RNA instead of DNA. Next slide. Sorry, please, you can. Change to the next one. Hello. Okay, next. Yeah, thank you. So the last example we are going to be considering is real-time PCR. Like the name implies, if I'm doing a text message, I'll type my message, I'll send my message to you, right? And then if MTN decides to say, or Glo decides to say there's no network, you will not see my message till maybe next tomorrow when the network gets correct, Sabi? So, but if I'm doing a WhatsApp message to you and you are online, and I'm online and I'm typing a message, you are going to see that I'm typing a message before I send the message. And once I send the message, you receive it instantly, Abi. So that's how I would explain or differentiate between real-time PCR and conventional PCR. So real-time PCR is the type of PCR where you can see the PCR product in real time as it happening. You are seeing whatever is happening inside the setup. You are seeing the reaction happening, both the temperature profile changes, whatever is happening, you are able to view it in real time. It's different from conventional PCR, which is endpoint based, in the sense that, oh, I'm going to set up something in the cycler. After setting up, I'll bring it out. I'll set up gel electrophoresis. I'll allow the gel to run. Then I'll do size-based discrimination. I'll then start interpreting the result based on the size of the separation of bands that I've seen. In this case, as the PCR is running, you are seeing amplification happening. So if a reaction is going to take 2 hours to be complete, and after 30 minutes, there's actually amplification on a particular sample, you are able to know that this sample is amplifying and is going to be positive. So real-time PCR is a very powerful tool in research, is a powerful tool in diagnosis, is a powerful tool in disease monitoring, and is a powerful tool in quantification, you know. So you are able to detect PCR product in real time. It's also known as quantitative PCR, or the one we say qPCR, and it requires the use of DNA binding dyes. It's used to measure the quantity of a PCR product, and it quantitatively measures the starting amount of DNA, cDNA, or RNA in your sample. So with this real-time PCR, we can quantify. You know, in our normal conventional PCR, it's just going to tell you, oh, this DNA of this so-and-so organism is present in this sample. This one is not present. But in real-time PCR, apart from the fact that you are able to understand or see the PCR product as amplification is happening, you are also able to quantify the starting material. So if I'm going to do, for instance, a viral load analysis, I want to check what the viral load is on this sample, then real-time PCR is the best bet, or quantitative PCR. Because apart from viral load, I can also use the cycle threshold value to monitor progress a patient is making during treatment. So if, you know, the topic of this module is actually PCR and its application in molecular diagnosis and monitoring. So we are not just interested in diagnosis, but monitoring. Let's say a person comes for test and you have a particular value, you have quantified the DNA, you have a particular value, and then this person goes on treatment for say 7 days, 21 days, or whatever, and then another sample is sent to you. You should be able to tell whether there is an improvement, you know. So this is where real-time PCR comes in. Next slide. Okay, so I want us to imagine running a real-time PCR. If you can zoom into this picture, this image, if you can increase the size of this image on your screen, you can look at the, okay, yeah. Okay. Please, it's too high. It's too high. Can you come down a bit, sir? Okay. A bit more down. All right. So what you can see here are PCR tubes having three different concentrations of, let's say, a particular organism, Abi. The green has 4 units, the red has 1 unit, and the blue has 1/8 units, right? So when you do a real-time PCR, for instance, of this particular sample with these units, this is what you are going to see. You can see the green, red, and then the blue clearly separated. So a line, or what we call the cycle threshold line, that we emanate from this part that has 5,000 down here, from the 0 point here, cutting across this is what will give us this cycle threshold value, 23, 25, and 28. So real-time PCR is what can give us this kind of differentiation in quantifying a sample. Next slide. Okay, so this slide is just buttressing what we have already said, showing us the difference between qPCR, that's the real-time PCR, quantitative PCR, versus endpoint PCR, or the one we call conventional PCR. So you can go through this when we are done. Next slide. Okay, so the next thing we'll look at is PCR controls. Yeah. So, we talked about how sensitive this technique is. We talked about the fact that little amount of contaminating DNA can give you a false positive result, can have ambiguous result. Even right from sample packaging, putting 2 samples, 3 samples together and sending to the lab, you can have a highly positive sample among those samples, even if it's not highly positive. And then there's a leakage and it just touches the cap of the other samples. It can actually give you false results. And we talked about how you can use your sample acceptance and rejection criteria to actually correct all of that. Apart from this, there are several controls that we incorporate into every PCR run that we do. So these are listed on your screen. Positive control is there, the negative control, negative extraction control, and then what we call the internal control. I'm going to take them one after the other. So for a positive control, I have my reagents, right? And then I set up a PCR. I run the PCR reaction. And at the end of the day, I don't have a single positive sample. And if there is no positive control in that particular run, how am I sure that my reagent is actually working? So positive control will help us to establish confidence that the reaction can actually amplify what it was designed to target. So positive control are actually templates from the exact organism that you are targeting. So if your positive control is set up in a PCR and it refuses to amplify, then you cannot say any negative sample in that run is negative. You won't have the confidence to give it out as negative. Same as negative control. I have my reagents and then I set up reagent and my reaction. And I put, let's say I just use distilled water or nucleus-free water to do a run, a setup. And if it's amplifying, then any other thing that is positive on that batch, it could also be amplifying based on the fact that my reagent can amplify anything. So I'm not going to take it as a true positive. So these are controls. Then the negative extraction control. I have the extraction process running. We've talked about the different kind of extraction, the spin column base extraction, the magnetic bead, and then we looked at the automatic extraction and procedure. So if I'm doing extraction, apart from the fact that I'm putting the samples in the tubes for extraction, I'm going to have a tube where I'm going to put nucleus-free water or something without DNA or RNA templates. And at the end of the day, it will help me monitor my extraction process to be sure that it is not spreading across that setup that is making my samples to amplify. And then what negative extraction control can also do for me, I can do a positive extraction control as well as a negative extraction control. So if I do a positive extraction control, remember I said the membrane binds with the nucleic acid material, the cellulose membrane that is part of the cellulose membrane that is part of that spin column. I said it will bind and form a bond with the nucleic acid material. And at the end of the day, when you are doing elution, it is expected that all of that nucleic acid material that has bind to that membrane will come down as your final template that you are going to use to do the setup. So if I do a positive extraction control, the positive extraction control should be able to bring out all of those templates that have bind to that spin column that were initially bound to that spin column. So if I'm not getting the exact concentration of the positive control that I use to set up positive extraction control, how am I sure that the template of the organism was also able to elute completely when I was doing my final elution? Then the last one I'm looking at is the internal control. Okay, yeah. So the internal control, or what we call the IC, in every setup, the samples that you have, the negative control, the positive control or negative extraction control, and internal control is like a standard check for every PCR reaction. So this one helps us to monitor PCR inhibition. Remember when we were talking about the quality of sample and extraction, I said DNA polymerase has so many things that can inhibit it, including the EDTA that is part of your reagents that you use for lysine, including the reagent that you use in even doing the extraction, the wash buffers. Most of those things have the ability to inhibit PCR. So for you to be sure that all of these have been excluded completely after extraction, that is how why the internal control is very important, or the IC. So the IC will help me to check for PCR inhibition. So IC must amplify. So if the internal control does not amplify, I cannot say a reaction is negative. So it must amplify with every reaction that I set up. Next slide. Okay, so this is considering applications of PCR. We have actually talked about several areas we can amplify it, we can apply it on, like in research, disease diagnosis, infectious disease diagnosis in agriculture, and several other downstream applications. You know, we talked about mutation-specific PCR. You can detect several mutations with PCR. You can detect infectious disease. You can help in disease monitoring. A patient that has been diagnosed, you can help to check if there is improvement. So molecular epidemiological studies is part of the application, monitoring the emergence and widespread of drug-resistant pathogens. I use the M. tuberculosis as an example, where I said when you are doing multiplex PCR, you can actually check if this organism is resistant to several antibiotics or if it's multi-drug resistant. And then molecular identification and detection of diseases is there. Gene cloning for sequencing is there. Next slide. Next slide, please. Hello. Next slide. Hello.
Over.
Hello. Okay. It's like the network locked you
Can
up.
you hear me? Yes. Can you hear me?
Okay, so you can share the next slide. Let's round up so that if there's any question, we'll quickly attend to it. Okay, so yes, we have looked at PCR, we have looked at the applications, the procedures, and everything. And you can agree with me that it's a very powerful tool and a very useful technique. So, but you know, everything that has advantage, sometimes they will say there is a disadvantage, right? Yeah, so does it have disadvantages? The answer is yes. The major disadvantage of this technique also comes from its major strengths. Like we have said, it can amplify few copies of DNA, and you have several thousands of copies of DNA, or millions of copies, or billions of copies of DNA from your singular sample. So small contaminated DNA can also amplify and give you false positive results. So that's an advantage, a disadvantage. And for the fact that most of these things we are saying are not cheap, the equipment, the training that is required for people to run this, experiment is actually not cheap. It is very expensive to carry out, and then it is highly sensitive. So the oversensitivity sometimes can also be a disadvantage. So at this point, if there is any question, please you can ask, and if there is none or any question later, you can post it on the group for us to attend to it. Thank you.